Next speaker is Peter Lansbury, who's been in drug discovery for quite a while. His picture's not, the name with his picture is not correct.
Peter Beard? Is he here? I'm not him.
Yeah, and he'll be speaking on the glycosphingolipid pathway, an introduction.
Okay, I need to shut this. Can I get some help, Zeev? While Zeev is taking care of this, I just want to thank, this is already, I can tell it's going to be so much fun, so great. And Lorna, where's Lorna? Has done an unbelievable job of shepherding us through this. I also want to thank Zeev because it's really great to have someone, a friend, invite you to talk about something that, a passion that the two of you share. And I wanted to say we don't always agree about that passion. That passion is NBA basketball. And so I brought some emails. So the title of this email, and this was on the neuro email system, by the way, was SSSPTB beta SNP and SBT activity. So I was asking him, I say to Zeev, and this was on early June, I say, I fear Luca, and I'm irritated by Tatum, but Brown, Holiday, and White have been great. So what is Zeev's response to me as a responsible scientist? His response was, sorry, Peter, Mavs will win. So, to keep Zeev humble, in all things, I brought you this. To wear during the meeting. So you got to take, when people claim to know a lot about a subject, you really need to be careful. So I'm going to give a talk today that's going to have a lot of wild speculation. That's my warning. It's going to be all fact-based. Zeev asked me to talk about the pathway, and all we really know about the sphingolipid pathway is sort of analogous to a flat map of Montreal, where you know where the streets are, you know where the corners are. You have no idea where the traffic's going, right? And so I'm going to talk a little bit about what we do know and a little bit about what we don't know. The other thing to remember is, unlike the traffic system in Montreal, the sphingolipid pathway has evolved to be functional and adaptable, right? And so you could imagine, I mean, in Boston, we have a zipper thing that changes the lane width, and that's a sort of a smart highway, depending on which way the traffic is flowing. But this system has evolved, and that's really important when you're thinking about trying to manipulate it, right? Because the manipulations you make are read by the cell and responded to. And so I'll talk a little bit about that, and I think it's really important, obviously, to understand. This is my version of Greg's, it's supposed to be simple slide. This is Dmitry Kranich called me in 2012 to get me involved in the company he was starting, Lysosomal Therapeutics. And I thought it was really easy. I remember telling my wife, I said, this is a piece of cake, okay? So, now how do I advance these?
This is your PDF, so just scroll.
Scroll? How do you, where? Oh, here? No. This is not an apple? Yeah. Okay, just click down there? Okay, there we go. So this is how I think of this now. It's a lot more complicated, and how PD drops out of this, we really don't know. But this is what I want to talk about, which is that GCase is part of this massive system that recycles sphingolipids, and we'll talk about how that works. Why is that important? It's important because the composition of sphingolipids has to change constantly. It has to be refreshed, and it has to change to adapt to things. It adapts to microbiome-derived sphingolipids. It adapts to dietary sphingolipids. There's been a lot of really good studies done on this in yeast, where, of course, everything's a lot easier, but it's very true, and the sphingolipid composition determines the membrane properties. I want to talk a little bit about it. Greg has already foreshadowed this, how membrane properties can go back and help regulate sphingolipid degradation, and in fact regulate GCase. So this is a self-correcting system. It's made to be flexible. It's made to be resilient to a lot of polymorphisms. And it affects sort of everything that we're going to talk about this this next few days, all of these organelles are affected by their membrane permeability, membrane fluidity, et cetera. This is unbelievable. Steve, would you just sit here and do this for me? How is this not working? I'm right there. Show me where that is.
Just above.
God, my kids will get a big kick out of this. Okay, so here's one example. I think this guy in this paper had a really great idea. So, the elephant in the room in every PD meeting is synuclein, and let's get synuclein out of the way. And say we don't know whether synuclein is an epiphenomenon, a linked epiphenomenon, or a causative agent. But it could be linked. This person links it very well. In this case, he was studying phospholipase A2 mutations that affect the chain length in these lipids. The chain length, in turn, affects curvature, membrane curvature. Membrane curvature affects synuclein binding, and synuclein binding to membranes is known to play a role in essentially inhibiting aggregation. So maybe the aggregation of synuclein has to do with membrane changes. It's just possibly an innocuous reporter of a membrane change. On the other hand, maybe it's self-driving. But I think this is a really interesting way of thinking about this. Oops. Okay, so here's a structure of a sphingolipid. This is ganglioside GD1A. This stuff is very polar and very specifically assembled, is present in the lumen or in the cytosol, depending on where it's located. This piece, which is the sphingolipid piece, is buried in the membrane, and that's really important to remember. It has two parts to it. One is the sphingosine chain, which can vary with respect to even insertion of a double bond is absolutely critical and has a really important effect on membrane fluidity, on lipid packing, and the acyl chain here. Now, I won't talk about the fact that there are maybe 10 major glucosylceramide isomers. They're all very different. Greg's work a long time ago showed that those chain links are differentially processed by GCase. So the chain length makes a difference on how fast that GCase reaction is. It also has an effect on how the membrane responds. So here are some of the features of membranes that we know are important. They're all affected by sphingolipids. I'll talk a little about curvature. Fluidity is really important. Permeability, of course, is really important. Membrane thickness is extremely important, and it's driven by the size of that ceramide piece. So the longer piece you get thicker membranes. Thicker membranes have different properties than thinner membranes, and there are many, many examples of that. So here's a version of GCase bound here in a model that was proposed, not by me, to a membrane surface in which glucose is embedded, and there's the glucose head group right here. And as Greg mentioned, I've left sapacin C out, but sapacin C is a critical component of this membrane-bound complex. The membrane itself has a huge influence on the activity of GCase. So in blue are wild-type enzymes. These are just in vitro assays that we put together at LTI. You can see without the membrane in blue, the wild type, and in the presence of membrane, you get a large increase of activity. You also get an increase in the affinity of the complex for sapacin C. And in the case of mutants, that effect is also there, but is smaller. So it's important to understand that GCase activity impacts membrane structure. Membrane structure impacts GCase activity. That's how you would set it up if you were, Mother Nature. But we ignore that kind of stuff too much, right? So here's the kind of thing, I've been thinking about. If you have different membranes, the same protein associated could have different activities. And, I could show you 20 papers from Greg that talk about this, and we experience this too at LTI. And so maybe the difference, between having enough GCase activity and not enough GCase activity might have to do with some sort of membrane abnormality. And we also know this is biophysics done, showing that the role of curvature is really important there. What about a non-optimal GCase? Well, maybe N370S, for example, which we know has low penetrance, is only a problem if the membrane can't adapt, right? It would want to adapt, right? That's the natural, but of course, when you adapt one part of your metabolism, there's prices that you have to pay, right? This is like traffic. And maybe the adaptation is a resilience mechanism. So maybe that's what allows you to get fully active GCase out of an intrinsically less active enzyme. And so I think we need to think about these things. One thing, of course, I keep talking about Greg because he always says the best things, but he was talking about this yesterday in our meeting last night about membrane changes. So we know there are changes in membrane properties in Gaucher cells, right? Cell surface membrane properties. And we're starting to know that in GBA Parkinson's as well. These have to do with, this is a fluidity measure. These are measures, other types of measures. Even cell shape could be changing. And the reason I'm emphasizing this is I think these are potential biomarkers. There's a group in San Francisco, Steve Finkbeiner, that's working with Google to just look at cell shape, right, using AI to take into consideration everything about cell shape. And they're finding really interesting differences in cells from people with various neurodegenerative diseases. These are all driven by membrane, okay? So here's the roadmap, the static roadmap, where everything starts with ceramide in the ER, later in the Golgi, and it can be derivatized in one of three ways: by galactosylation, glucosylation, or conjugation with acetylcholine. And then these can be further elaborated in the case of the glue pathway to produce a gangliosides. I showed one complex one later on. Later on, these are transported to lysosome and sequentially degraded using a series of hydrolytic enzymes, most of which are membrane associated. So the whole machinery to drive this pathway is attached to the thing that it's maintaining. It really is kind of beautiful if it weren't so aggravating and complicated. Now, what is the purpose of this? No one knows. I don't know. My speculation is that it's important to keep enough ceramide, the supply lines of ceramide coming, because sometimes you need to change from GalSear to GluSear, right? Something happens, you need to change. And when you need to change, right, the only way to change is to digest and start again. There's no editing of sphingolipids, right? So you have to start back at the beginning. So you have to have this active system to digest and start again. And that's my speculation. We could talk over some beers about this. What do we know about GBA-PD and that idea? We know two things. First of all, if GCase is slow, for whatever reason, that appears to increase the risk and the rate of progression of Parkinson's disease. That's why we're here. So slow GCase would slow the rate of sphingosine production in the lysosome, slow the rate of replenishment of cytosolic ceramide, the building block. The other thing we know comes from the Sanofi trial of venglustat is that if you inhibit this pathway, GC synthase, in N370S-PD, the data is very clear that you accelerate progression of the disease. That's also consistent. So slowing down this pathway actually promotes Parkinson's disease progression in some small way. Okay, that's my contention. This is the only data we have so far. Now, I want to talk about another part of regulation. Aside from that, this sort of vague sort of membrane thing, there's specific things that can be done. And this is, now I've added pieces to this pathway. And these are the synthetic piece, the only entry to the pathway comes over the de novo synthesis pathway, over the bridge, right? And so this pathway is the only way we can make ceramide. And this pathway is the elimination pathway, the only way to get rid of sphingosine. So it's sort of a closed system. And sometimes, if you don't have enough ceramide, you may need to turn this on. This pathway is normally turned off, which I find amazing, that there's this incredible complex metabolic pathway that's typically off, right? It's off because ceramide itself keeps it off. If ceramide levels drop, this pathway turns on. Okay? And so maybe this is something important, and this is something that I've been really interested in lately. This is the enzyme complex called serine palmitoyl transferase that starts this de novo pathway. This is the structure in the inactive form. Okay, so activation requires a dissociation of these purple pieces and then activation. There are these are regulatory subunits, these green helices, and this heterodimer contains the active site. There is a SNP in the regulatory subunit that's linked to PD risk, and it's pretty robust. That I find very interesting. There are also mutations in this that are linked to hereditary spastic paraplegia. There is a spontaneous mutant mouse that Jackson Lab discovered called Stellar that exhibits rapid neurodegeneration. It happens because of a mutation in the regulatory subunit. So this suggests to me that regulating the production of new ceramide has some importance in these diseases. And so this is really a crude thing that we put in a paper when we showed our data from our phase 1b trial to try to understand this. And so it might be that if you have low flux here through this pathway, you supplement it by turning this pathway on, and you really don't want to do that, right? It's there because you need to activate it in emergencies, but you really don't want to do it. There are negative consequences of that. And so there is a real big question when you switch from here to here, for instance, by adding more GCase, by gene therapy, let's say, how does the cell respond to that? And one thing that's really interesting to me is all these things are time dependent too. So one thing that people in our field rarely do is do time courses of responses and things. We saw a really, really interesting time course of response in our phase 1b trial that I'm comfortable with at this point. So how do we turn this piece into a biomarker? So this is some work that I'm really passionate about, and no one will pay me to do this, but including BL actually. The idea is instead of trying to study traffic by taking aerial photographs of Montreal and saying, oh, it's crowded at this corner, it's crowded at this, we're going to do what Waze does, which is it tracks each individual car in real time, that's a member of Waze, and it then can intuit, or not intuit, but AI the way to a traffic flow. So we need a way to track the flow here. And the way we're doing this is with isotope incorporation. So we isolate, this is an assay that we worked out to be used in the clinic. And so this could actually be applied to a large clinical trial. The CVs are very low. And what we're doing here is we're, in this case, incorporating a labeled serine so we can track from the time that that serine is added, we can see now an increase, in this case in glucir with label, and in this case in sphingomyelin with label. These are stable. This interpatient difference is real. We know that because before we did this was on 24 patients with and without PD. We did a lot of patients looking throughout the course of the day it's stable. If I come back next week, I can make glucir a lot faster than Roy, and we don't know why. But there are differences that can be measured. And if one wanted to modify this, this is a potential tool to actually assay the de novo pathway in vivo. So, yeah, I just want to stop and thank you for putting up with my slide problems.